This is the lecture for Chapter 3, Organic Molecules. As I mentioned in a previous lecture, science tries to organize the world in a logical order, and chemistry focuses on that order on a small level, on the atomic and molecular level. More specifically, chemistry is focused on the subatomic particles, how they go together to form atoms, and how those atoms come together to form molecules, and how those molecules come together to form macromolecules. Now, there are two types of chemistry. Inorganic chemistry deals with non-living things, and non-living things, or inorganic chemistry, tend to be composed of a lot of ions, which are held together by ionic bonds. Those molecules tend to be very small, and of course those are molecules you do not find in living things. Organic chemistry, on the other hand, has to do with the molecules that you find in living things. And these molecules tend to have a lot of carbon to hydrogen bonds. They are composed or held together by covalent bonding, and these molecules are very, very large. Because this class is a biology class and we focus on the structure and function of the cell, we are going to go over organic chemistry. Now, with organic chemistry, these are the four types of elements that make up the majority of the organic molecules that you find in living cells. And you should become very familiar with the atomic structure of these four atoms. So remember atomic structure. We basically have the electrons, the protons, and the neutrons. The electrons are orbiting around the nucleus, which is composed of the protons and neutrons. So the first one, hydrogen. Hydrogen has an atomic number of 1, which means it has 1 proton in the nucleus, and it actually has no neutrons, and it has 1 electron orbiting around. So it had in the first orbital, so it has a valence number of 1. Oxygen has the atomic number of 8, so that means that it has 8 protons and 8 neutrons in the nucleus, and it has 8 electrons orbiting around it. So it has 2 electrons in the first shell, so that is complete, and then it has 2 pairs in the second shell, and then 2 unpaired electrons in that shell. So it is looking to form 2 covalent bonds with those 2 unpaired electrons in the valence shell. Nitrogen has the atomic number of 7, which means it has 7 protons in the nucleus and 7 neutrons in the nucleus, and it has 7 electrons orbiting around that nucleus. So the first shell is filled, the second shell has a pair of electrons, and then 3 unpaired electrons, which are ready to make 3 covalent bonds. Carbon. Carbon is a very important atom for organic chemistry, and carbon has the atomic number 6, which means it has 6 protons and 6 neutrons in the nucleus, and then it has 6 electrons orbiting around that nucleus. So of course the first shell is filled with a pair of electrons, but then in the outermost shell you have 4 unpaired electrons. So those 4 unpaired electrons are available to make 4 covalent bonds. When talking about organic chemistry, you need to know a lot about carbon, because organic chemistry, those organic molecules, sometimes referred to as macromolecules or biomolecules, they are based on carbon. And that's also why sometimes maybe you've heard that living things are carbon-based organisms, or life on Earth is carbon-based. So carbon is a really important atom with some unique characteristics, which makes it perfect to build these different types of biomolecules. So again, going over the structure of carbon, carbon has the atomic number 6, which means it's going to have 6 protons and 6 neutrons in the nucleus. So its overall atomic mass is about 12, because you count the mass of the protons and the neutrons, not the electrons, because they weigh so much less comparatively. So you have 6 protons, 6 neutrons in the nucleus, and then you have 6 electrons orbiting around the nucleus. The first shell is filled with the pair of electrons, so that is filled, and then you have the second shell which has 4 valence electrons. So 4 valence electrons, 4 unpaired electrons. So these electrons are looking to form a covalent bond to fill out their pair. So the thing that makes carbon unique is that it's a relatively small atom, so it has a small nucleus, so only 6 protons and 6 neutrons, but it has this ability to form 4 covalent bonds. And covalent bonds are very, very stable, especially when you have carbon-to-carbon covalent bonds. So carbon is able to, each carbon atom can form 4 individual single covalent bonds. And this is what you see in ethane. You see 2 carbon atoms, and each of them has formed 4 covalent bonds with another atom. But carbon can also form double bonds. So instead of sharing 1 pair of electrons, it can share 2 pairs of electrons, and carbon can even form triple bonds where you share 3 pairs of electrons. Now, as more pairs of electrons are shared, so as you go from a single bond to a double bond to a triple bond, the bonds get stronger, they get shorter, so those nuclei are brought closer together, and they're more rigid. So when you compare a single bond to a triple bond, the distance between these 2 nuclei is going to be a lot closer, the bond's going to be a lot stronger, and it's going to be more rigid. There's going to be less flexibility. So part of being able to form single bonds, double bonds, triple bonds between carbon atoms gives carbon an ability to form a variety of different structures. As I mentioned before, life on Earth is considered carbon-based, and that's carbon-based because all the macromolecules, all the biomolecules that make up living things are based on hydrocarbon skeletons. So hydrocarbons mean hydrogen bound to carbon. That forms the framework on which all our macromolecules are based. And with these hydrocarbon skeletons, you get a lot of variety. Now, I already mentioned about the bonding. So carbon can form single bonds and double bonds, and depending on where those double bonds are located, that can influence the flexibility of the molecule. Now remember also, carbon can also form triple bonds. But also you get differences in structure with the length of the hydrocarbons. So the length of the hydrocarbons, they can be very short, just 1 or 2 carbons long, or they can get longer. That one, propane, shows 3. Or you can have molecules where you have thousands of carbons long. The hydrocarbon skeleton is thousands of carbons long. Also, carbon can branch because each carbon atom can form 4 covalent bonds. You can get all sorts of branching, and it can become very, very complex in the molecule. And the last type of variety are rings. So hydrocarbons can form into rings, and depending if all the bonds are single bonds or you include double bonds, that can affect the function of the ring. It affects the flexibility. So carbon, because carbon is a relatively small atom and it can form 4 stable covalent bonds to other carbon atoms and also to hydrogen and other atoms, you can get a wide variety of different molecules in terms of shape and function. In addition to the hydrocarbon skeleton, all the different types of biomolecules have functional groups attached to that hydrocarbon skeleton. And the functional group is a specific group of bonded atoms. So it's a group of atoms, particular atoms bound together that always react the same way. So they have certain chemical properties. And this is what's going to determine the specific characteristics of the different types of biomolecules. And this table just shows you a few of the different types of functional groups. And when you're looking at this, for instance, for the alkane, R refers to the hydrocarbon skeletons. So in this case, the functional group is actually composed of 2 carbons and 4 hydrogens bound between 2 hydrocarbons, 2 carbon skeletons. For instance, alcohol here. Alcohol is an oxygen and hydrogen that is bound to a hydrocarbon skeleton. Now, more specifically, I'm going to go through 7 of the most common functional groups that we will actually be talking about today as we go through the different types of biomolecules or macromolecules. So this is a table. It gives you the structure of the functional group, the name of the compounds, what compounds it's found in, an example, and then the functional properties. So the first functional group, which is very, very common, are the hydroxyl group or a hydroxyl group. And this is an OH. So it's an oxygen bound to a hydrogen. And then at the end of the dash is the hydrocarbon skeleton. And for this, usually this is found in alcohols. So if you see a molecule that's name ends in OL, it is probably an alcohol, and it has an hydroxyl group bound to it. And this is just an example of ethanol. That's the type of alcohol that we drink, and that's what it looks like. And the functional properties of a hydroxyl group is that it is polar. So this is a polar, and it can hydrogen bond with water. So it can dissolve in water, and you often found this on sugars. So any molecule that is a sugar often has a hydroxyl group bound to in its structure. Second is carbonyl group. A carbonyl group is a carbon bound, double covalently bonded to an oxygen, and then it binds to a hydrocarbon skeleton on each other side. And there are 2 types of these, the ketones and the aldehydes, and it depends where you find the carbonyl group. If it's in the middle of the molecule, it's a ketone. If it's at the end of a molecule, it's an aldehyde. And these are important in terms of isomers, which I'll talk about in a minute, but you often find these in sugars. The last functional group on this slide are the carboxyl groups. Carboxyl groups, this is a carbon bound to 2 oxygens and a hydrogen. So it's written like this structurally, and then in terms of formula, it's written like that. And this is part of an acid. So when you see this group on a molecule, you know it has acidic properties, which means it can increase the hydrogen ion concentration when it's dissolved in water. And this is what you see in vinegars. So vinegars, hopefully you remember, vinegar has a low pH. Okay. This slide has the last 4 functional groups that I want you to know. And on this slide, the first one is the amino group. So the amino group has 1 nitrogen and 2 hydrogens bound to it. And the amino group is found on amino acids, and amino acids are the building blocks for proteins. So this will be important when we're talking about the proteins. Also, amino groups act as a base. So when you dissolve them in water, they can pick up hydrogen ions, so they can increase the pH of a solution when they're dissolved in the solution. Second group are the sulfhydryl group, and that has a sulfur and a hydrogen. And this is often found in proteins. So this group is found in proteins. It's found in certain types of amino acids, and it's important because it can cross-link to another sulfhydryl group, and that will stabilize the protein structure, which, as you will see, will be very important to protein function. The next group is the phosphate group. Phosphate is a phosphorus phosphate with 4 oxygens bound to it, and the important thing here is it's going to have a strong negative charge. So those oxygens are going to have a negative charge, and this is going to be important. This phosphate group having this negative charge is going to be very important with ATP. The function of ATP is the readily usable energy of the cell. And phosphate groups are also found in the nucleic acids, the DNA and the RNA. It's part of making the backbone for the nucleic acids. The last group are the methyl, or is the methyl group. Methyl is 1 carbon bound to 3 hydrogens, and the methyl group is really important to DNA. So you can find methyl groups attached to certain DNA molecules, and that affects the expression of genes. Basically, it stops the expression of certain genes, it turns them off. And also the methyl groups can be related to sex hormones, and it can affect their shape and function. Another thing about the organic molecules is that they can have different isomers. And iso means the same, and these are molecules which have the same molecular formula but different arrangements, and those different arrangements give them different functions. So with these isomers, first type of isomer are structural isomers. And structural isomers have the same types of atoms and the same number of atoms, but they are bonded together in different structures. So an example could be pentane and 2-methylbutane. The pentane has 5 carbon atoms bound together in one chain, and surrounding that are the 12 hydrogens that are covalently bonded to the carbons. 2-methylbutane has 5 carbon atoms with single bonds bound together, but now it's in a branched structure. And even though it's a branched structure, you still have 12 hydrogen atoms bound to that carbon molecule. So pentane and 2-methylbutane have the same number and the same types of atoms, but they're bound together in different structures, and that gives them different functions. Second type of isomer are geometric isomers. So geometric isomers also have the same number and same type of atoms, but here they have a very similar structure. And in this case, you have 2 atoms that are bound together by double bonds, and then each carbon atom is singly bonded to a hydrogen and to some other functional group. Now, when you're talking about these functional groups, the X's represent the functional groups. You can have cis isomers and trans isomers, and that refers to the location of those functional groups. If it's a cis isomer, they're on the same side of the molecule. If it's a trans isomer, they're on opposite sides of the molecule. The last type of isomer are enantiomers, and enantiomers have the same, of course they're isomers, they have the same types of atoms, the same number of atoms, and with an enantiomer they have the same connectivity, but now the shapes are opposite. So the 3-dimensional shapes are opposite. So in this case, you have a central carbon, which has a covalent bond to 4 different groups. And this one also has the central carbon and single bonds to the 4 different groups, but now they have an opposite 3-dimensional shape. And when we refer to this, well, an analogy is like your hands. So your hands, on each hand you have a thumb, which is next to a pointer finger, then a middle finger, ring finger, and a pinky. And your right hand has the thumb with the fingers in the same orientation, but they are opposite to each other. You have opposite 3-dimensional structures with your hands. And so because we use this analogy with the hands, with the enantiomers, we talk about left-handed isomers or left-handed molecules, and then right-handed molecules. And for whatever reason, most of the molecules on Earth that interact with living things tend to have a left-handedness. So our cells are more accustomed to building molecules and dealing with molecules that are L isomers or have a left-handedness. Metabolism is an important characteristic of living things, and metabolism can be defined as the sum of all chemical reactions that occur in a cell or a living thing. So you can talk about a living thing as being composed of one cell or a multicellular organism. And in general, when a living cell or a multicellular organism is metabolizing, in general, what's happening is that organism is taking in food, taking in nutrients, and when that living organism takes in nutrients, it's going to break those nutrients down into smaller particles or subunits. And there are a couple of reasons why a living thing is going to take in food particles and break them down. One is to release energy, and that energy can also be used to increase the temperature of the living organism, or that energy can be used with those subunits to form new macromolecules. So in general, when we're talking about metabolic processes, there are basically two things that the living cell is going to do. It's going to take large molecules and break them down, and then it's going to take some of those subunits and put them together to build up larger molecules. Breaking down of large molecules is called catabolism, and building molecules up is called anabolism. All of metabolism basically encompasses two types of reactions, catabolic reactions and anabolic reactions. And this is an example of hydrolysis. And hydrolysis is a type of catabolic reaction. So it's where you are taking a large molecule, usually a polymer. And a polymer, poly means many. A polymer is a large molecule made of similar subunits covalently bonded together. So in a hydrolytic reaction, a hydrolysis reaction, what's basically going to happen is one of these subunits is going to be broken off. So this is a catabolic reaction where you're taking something large and breaking it into smaller subunits. Now with a catabolic reaction, this involves a molecule of water splitting. So we say its hydrolysis is water splitting because this molecule of water is going to split, I'm using quotation marks, into its two components, the hydrogen and the OH, and these are going to make up the covalent bonds that were broken, the covalent bond that was broken during hydrolysis. So what is basically happening in a hydrolytic reaction is that you have this polymer where you have these similar subunits held together by covalent bonds. So in order to break off this subunit, this covalent bond needs to be broken. So now these two subunits who were sharing a pair of electrons, they are no longer sharing a pair of electrons, and so each subunit has an unpaired electron. And in general, that is not a stable thing. So molecules like to have their electrons paired. And so in order to sort of repair these electrons, you need to reform these covalent bonds. And because metabolism, biochemistry, occurs in water, and I've mentioned before that water and liquid water, you have some molecules that just spontaneously split, where the hydrogen splits off and then the OH is left. So in water, you'd have this already split. So then the hydrogen is ready to form a covalent bond with that subunit, and the OH is ready to form a covalent bond with that subunit. So that's basically a hydrolytic reaction. Now, when you have hydrolytic reactions, when you are breaking these covalent bonds, often they're a carbon to hydrogen covalent bond, and that releases energy. So hydrolytic reactions tend to be exergonic. They tend to have a net release of energy. The other type of metabolic reactions are the anabolic reactions. And anabolic reactions are where you're building something larger. So this is where you're taking a short polymer and going to link a monomer subunit to it to create a longer polymer. So in order to do this, you need to break existing covalent bonds to allow these two subunits to then form a covalent bond to each other. And to do this, these are usually in organic molecules. Organic molecules often hydrocarbon skeletons, so somewhere on that molecule there is probably a carbon bound to a hydrogen. Somewhere on the subunit you have a carbon bound to an OH molecule, so if you break those covalent bonds, now that hydrogen and OH are free to form a molecule of water, and now these two subunits can form a covalent bond to each other. So this is often called a dehydration reaction, because when you dehydrate something, what are you doing? You're removing water. So that's basically the opposite of the hydrolytic reactions. Hydrolytic reactions, you're splitting a molecule of water. This one, you're forming a molecule of water. And these reactions are often endergonic. they require a net input of energy. So it takes energy to form, this bond. All the structures of all living things on Earth are composed basically of 4 different types of macromolecules. And the macromolecules are hydrocarbon skeletons with different types of functional groups bound to them. And the 4 types of macromolecules are the carbohydrates, the lipids, the proteins, and the nucleic acids. And I'm going to go through each one, starting with the carbohydrates. So carbohydrates, carbo stands for carbon, hydrate stands for water. So that basically tells you the types of atoms that are involved in these molecules. So they are mainly carbon, hydrogen, and oxygen. And the basic purpose of the carbohydrates in terms of living things is to provide immediate energy and structure. There are 3 general types of carbohydrates, and they are basically organized by size. So you have the monosaccharides, the disaccharides, and the polysaccharides. But no matter which group of carbohydrate a molecule belongs to, most carbohydrates have a particular ratio of atoms. And the ratio is 1 carbon to 2 hydrogens to 1 oxygen. And this is basically the ratio, and in order to find out exactly how many atoms of each type you have in a carbohydrate, you need N, which is the number of carbons. So for example, these are monosaccharides. Mono means 1, saccharide means sugar. These are the simple sugars. These are the basic subunits that most of the other carbohydrates are composed of. So in this example, you have an aldose. This is an example of an aldose. It is a 3-carbon monosaccharide. So if N equals 3, then without counting up the atoms, you know that the aldose is going to have 3 carbons, 6 hydrogens, and 3 oxygens. So that would be the molecular formula for aldose. And as you can see, this is true. It has 3 carbons, 6 hydrogens, 1, 2, 3, 4, 5, 6, and it also has 3 oxygens. So this holds true for most of the carbohydrates. As long as you know the number of carbons, you can figure out how many hydrogens and how many oxygens make up the structure of that carbohydrate. So these are an example of 5 different types of monosaccharides: aldose, ketose, triose, pentose, and hexose. And the first 3 have 3 carbons in their structure. Pentose has 5 carbons, and hexose has 6 carbons. And the hexoses, these are the most common monosaccharides that we will be talking about, and specifically glucose is probably the most common monosaccharide that we talk about in this class. This is an example of glucose. Glucose, again, is a monosaccharide simple sugar, and these are different ways to write the formula for glucose. And this is a linear way, but glucose in reality tends to form a ring structure. So this is usually how we refer to glucose, and often we don't add in all the different atoms and functional groups, and so we just denote it as this sort of green structure. These are other examples of monosaccharides. So glucose, like I mentioned, this is the most common one we'll probably be talking about. This is the blood sugar. This is what your cells break down to produce energy. Fructose is the sugar the monosaccharide that is most commonly found in fruit. And then down here we have deoxyribose and ribose. These are the 2 types of sugars that you find in nucleic acids. The second type of carbohydrates are the disaccharides. Di means 2, saccharide means sugar, and this is where you take 2 monosaccharides and bind them together in an anabolic reaction to produce a disaccharide. So this is an example of a dehydration reaction forming a disaccharide. So here is a glucose molecule, here is another glucose molecule, and in order to link them together, You need to break the covalent bond between this carbon and this oxygen, and also break the covalent bond between the oxygen and the hydrogen. When you do that, now the oxygen and this hydrogen can bond together to form a molecule of water, hence forming a molecule of water, dehydration reaction. So now that those covalent bonds have been broken, now this carbon is free to form a covalent bond with that oxygen from this molecule. And again, dehydration reaction, this is an anabolic reaction, so it requires an input of energy. Anytime you're building something up, creating a larger molecule, it tends to require a net input of energy. This is another example of a dehydration reaction. This time it's between a glucose and a fructose. Again, you're going to break covalent bonds between that oxygen and that carbon and break the covalent bond between the oxygen and hydrogen, and then that will result in the formation of a molecule of water. And now you are forming a bond, that covalent bond, which is called a glycosidic bond. Anytime you are linking monosaccharides together, we technically call that a glycosidic bond. Now that you have glucose bound to fructose, the resulting disaccharide is called sucrose, and sucrose is table sugar. One of the things you should have noticed is that sugars tend to end in the suffix -ose. So pretty much anytime you see a word that ends in -ose, it is some kind of sugar. The last general type of carbohydrate are called the polysaccharides. Poly means many, saccharides means sugar. So these are incredibly large molecules that are made of simple sugars bound together with those glycosidic linkages. And some examples of polysaccharides are cellulose, starch, glycogen, and chitin. This is a table that compares a couple of different types of polysaccharides to each other. So this is cellulose, two different types of starch, and then glycogen. And the first thing is to point out where you actually find these different types of polysaccharides. Cellulose is found in plants, and it usually holds the structure of the plant. Starch is also found in the plants, and this is the energy storage molecule. Glycogen is actually a polysaccharide that animals produce, and that is also used for energy storage. But if you look at this, they are all composed of glucose, slightly different types of glucose, and the structures are different. So if you look at the shape, the structures, they're a little bit different, and that gives each type of polysaccharide slightly different characteristics, because structure is very important to function. One example of polysaccharide is cellulose. And as I've mentioned before, cellulose is produced by plants, and it involves these long polysaccharides, long chains of monosaccharides bound together, thousands of monosaccharides bound together to create the cellulose molecules. And even those molecules are then twisted together to form microfibrils that form the structure of plants. Now, the interesting thing about cellulose is because it has a slightly different structure than starch, animals are unable to digest cellulose. So we can digest starch that you find in potatoes, you find in grains like flour. We can digest that and we can use that as a source of energy. But cellulose, our cells are unable to digest it because of the structure of how those glucose molecules are bound together. A slightly different type of polysaccharide is called chitin, and chitin has a different structure because it contains a functional group with a nitrogen in it. So it has a little bit of a different molecular formula compared to the other types of polysaccharides. But chitin is found in the cell walls of fungi. So all the mushrooms, their cell walls are composed of chitin. And also, chitin is found in the exoskeletons of insects, spiders, and also lobsters and shrimp. And the interesting thing about chitin is that also similar to cellulose, we are unable to digest it. So our bodies are not capable of digesting chitin. The second group of macromolecules that I want to discuss are the lipids. And the main characteristic about the lipids is that they are nonpolar. They do not dissolve in water. Carbohydrates are able to dissolve in water, especially the mono and disaccharides. Polysaccharides are less likely to dissolve in water because they're so long, but they are still hydrophilic. They can still attract water. Lipids, on the other hand, they are all nonpolar. They are all hydrophobic. And there are a couple of different types of lipids. You have fats, oils, phospholipids, steroids, and waxes. And depending on their type, they have slightly different function. Fats and oils are for long-term storage. Fats relates to the animals, and oils relates mainly to plants. Phospholipids are an important component of the plasma membrane, so the structure of the cell membrane. Steroids are a type of hormones that's used in communication between cells, and waxes are mainly for protection. And in plants, it's to protect from water loss. So those are some of the characteristics or the functions of lipids. Lipids are also unusual because they do not have the basic monomer polymer structure. So the different types of lipids have slightly different structures, and they don't follow that monomer polymer type of structure that most macromolecules have. So in this slide, you're going to see an example of a triglyceride. A triglyceride is the basic structure for the fats and oils that you find for long-term storage in animals and plants. So a triglyceride has two basic components. The first is a glycerol. Glycerol is an alcohol because you have these hydroxyl groups bound to it. So it's a 3-carbon alcohol. Then to form a triglyceride, it would add a fatty acid to bind to each carbon. So fatty acid, it's an acid because it has a carboxyl group. So for this, forming an ester linkage, that would be an anabolic reaction, a dehydration synthesis reaction. So the ester linkage is linking these fatty acids to each of those carbons. So triglyceride has 3 fatty acids, 1 coming off of each carbon. And with these triglycerides, what you want to notice is that triglycerides are often used for long-term energy storage, and that there are lots of carbon to hydrogen bonds in the triglycerides. So that means they actually have more potential energy than a carbohydrate. So often about twice as much potential energy. Because remember, think that the energy in a molecule is in that carbon to hydrogen bond. So with triglycerides are a glycerol, 1 glycerol bound to 3 fatty acids by an ester bond, ester linkage. With those fatty acids, there are 2 types of fatty acids. There are saturated fatty acids and unsaturated fatty acids. First type, the saturated fatty acid, this means when you look at the carbons in the fatty acid chain, that all those carbons are linked together by single bonds. So there are only single bonds between the carbons, which means that you can have the maximum number of hydrogens bound to those carbons in the fatty acid. So for example, if you have only 2 carbons in this fatty acid, and this is to the rest, this is the bond to the glycerol, then you can have a maximum of 1, 2, 3, 4, 5 hydrogens. So all of those would be hydrogens. So 5 hydrogens. So you've saturated the molecule, or the molecule is saturated with the maximum number of hydrogens. When you have only single bonds in a fatty acid, then you have a very sort of flexible fatty acid that can be packed in very tightly with other triglycerides. So what this means in terms of characteristics of the fat, these are usually fats. These are usually associated with animals, and a saturated fatty acid or saturated fat will often be solid at room temperature. If you compare that to the unsaturated fatty acids have a double bond between carbons. So somewhere in that fatty acid chain, you have a double bond between carbons. And if this is a very short fatty acid attached to the glycerol, this is called unsaturated because you're not going to have the maximum number of hydrogens. So in this case, this carbon can only form one other covalent bond, and this carbon can form two other covalent bonds. So if you have this configuration, you're only going to get a maximum of 3 hydrogens. Whereas up here, you had 5 hydrogens bound to that. Now you only have 3. So that's where the term saturated comes from. So saturated, maximum number of hydrogens. Unsaturated, you don't have the maximum number of hydrogens because you have this double bond. All right, also with a double bond, because if you remember when I mentioned double bonds before, double bonds are shorter than single bonds and they're more rigid and they create kinks in the fatty acid. So now you have a kink in the fatty acid, and now the fatty acid cannot be packed in tightly with other fatty acids. So what this means for the lipid is that it tends to be liquid at room temperature, and unsaturated fatty acids are mainly associated with plants. You've probably heard the term polyunsaturated fat. That means there's more than one double bond in the chain. So instead of having just one kink in the chain, you might have another kink if you have 2 or more double bonds in the fatty acid. Another type of lipid are the phospholipids, and phospholipids are similar in structure to triglycerides because they start with the glycerol, the 3-carbon alcohol. But in this case, the phospholipid only has 2 fatty acids. So only 2 fatty acids have ester bonds to the glycerol, and then the other carbon in the glycerol is bound to a phosphate group. So that gives rise to the name phospho. So it's bound to a phosphate group. And this gives the phospholipids a very unusual characteristic. The phosphate group has a negative charge on it. So that means that the head, we call it the head, the part that has the phospho group on it, is hydrophilic. And then the tails are hydrophobic because hydrocarbons tend to create nonpolar molecules. So these are hydrophobic tails. So this molecule, the phospholipids, have a very unusual characteristic. One end is a hydrophilic head. The other side are 2 hydrophobic tails. The interesting thing about the phospholipids is when you mix them with water, they will automatically form spherical structures. And those spherical structures are composed of a bilayer. So the bilayer is organized with phospholipids by 2 layers, where the hydrophilic heads are interacting with water outside of the sphere, and the second layer, the hydrophilic heads are interacting with water on the inside of the sphere. And the nonpolar tails, hydrophobic tails, are organized together this way. And this is very important because this forms the basic structure for cell membranes. Steroids are another example of a lipid. And one example of a steroid is cholesterol. So this is the structure of cholesterol. And with the steroids, you see all these ring structures. And the other thing about the steroids, of course, is that they are also nonpolar. They do not dissolve in water. In a cell membrane, cholesterol has a very important role. It is embedded in that phospholipid bilayer, and this helps maintain the rigidity of the animal cell membrane. So in animal cells, you will have cholesterol embedded in the phospholipid bilayer. Cholesterol is also important in multicellular animals like humans because it is used to form testosterone and estrogen. And you should know that testosterone and estrogen are 2 different types of hormones that can have very different effects as an individual develops. And hormones, remember, are molecules, communication molecules, signal molecules that allow different tissues to differentiate in different ways in, of course, males and females. The last type of lipid are the waxes. This is the basic structure of the waxes, and the waxes, just like all the other lipids, are also nonpolar. But waxes tend to be made by plants mainly, and their purpose is to protect from water loss. So a lot of plants will have waxes on the leaf, waxy leaves, to prevent water loss and dehydration. Beeswax is another common use for waxes. So bees can produce wax, and that's basically for structure, holding the structure of the honeycomb for storage of the honey. And in humans can produce waxes. Earwax is the common one that we think of, and earwax is also protective. It can protect from infection in the ears. The next group of macromolecules that I want to discuss are the proteins. And proteins are very, very important for living cells, and they actually make up about 50% of the dry weight of a cell. So if you take out the water, 50% of what is left is composed of proteins. And proteins have lots of different types of structures, which gives them lots of different functions. And this table is an overview of some of those functions. So one of the main functions of proteins is to catalyze reactions, to act as enzymes. Also, proteins are involved in structure. So structure for the cell, structure for an organism. They can also store amino acids. They can be used to transport other substances around the body or around the cell. Some proteins act as hormones, and these are molecular messengers sending signals to different types of cells so they can respond. Also receptor proteins, these are the proteins that receive the hormones, receive the signal that allow the cell to respond. Also, proteins are involved in movement, muscles for a multicellular organism, and then actin and myosin to help move individual cells, and then defensive proteins. So defense antibodies are actually proteins, and they protect you from infection. One of the things I want to point out with all these different functions is that you don't see energy. So under normal circumstances, a cell will not use their proteins to produce energy. That is the purpose of the carbohydrates and the lipids. Those provide energy. Proteins do not under normal circumstances. Proteins do follow the classical macromolecular structure where you have monomers that bind together to form polymers. The basic monomer for the proteins are called the amino acids, and this is the structure for a basic amino acid. Every amino acid has a central carbon, often called the alpha carbon, and like every carbon, it is capable of forming 4 covalent bonds. One covalent bond will be to an amino group. The other covalent bond will be to a carboxyl group, and carboxyls, remember, when they dissolve in water, they form an acid. So that's where the name comes from, amino acid. The other covalent bond is formed to just a hydrogen, and the last one is to an R group. And to an R group, that is a functional group that will give that amino acid a particular characteristic. When 2 amino acids are linked together, they form peptide bonds. So with the dehydration synthesis reaction, here you have 2 amino acids that are already linked together, and you want to add a third one to it. So again, breaking these covalent bonds to the hydrogen and the OH here, this is going to form a molecule of water, and now a new covalent bond will form between this carbon and nitrogen. So that is a peptide bond. So again, proteins do follow the macromolecular structure of monomers linked together to form a polymer. In nature, there are many different types of amino acids as defined by their different types of R groups. But when you're looking at organic chemistry and the amino acids that you find in the proteins that make up living things, only 20 amino acids are found in living things. And these are diagrams of the 20 amino acids that make up living things, and they are grouped together by the characteristics of the R groups. So first you have nonpolar R groups. So these are the nonpolar R groups, and these are the examples. Glycine has just a hydrogen as its R group. Alanine has a carbon and 3 hydrogens, and then valine has this structure. And all of these, leucine, methionine, and isoleucine, all of these are nonpolar. Then there are polar R groups. Polar R groups are found on the amino acids serine, threonine, cysteine, proline, asparagine, and glutamine. You also have aromatic R groups. Aromatic means that they have a ring structure on them. So that's phenylalanine, tyrosine, tryptophan. Then you have R groups that can have a charge to them. So some that have a positive charge would be lysine, arginine, histidine, and some too have a negative charge, that's aspartate and glutamate. Now you do not have to memorize the different types of amino acids, but you should be aware of their names, because when we go through gene expression, it relates how the genes encode the information to make the different proteins, so the order of the amino acids. Now that's very important aspect of proteins is their structure. So we have 20 different amino acids, and with this we can get hundreds of thousands of different types of proteins. If you think of an analogy of language, in language we have 26 letters in the alphabet, and we can create hundreds of thousands of different words by using different numbers of letters and organizing them in different ways. And that's the same for the proteins. There are hundreds of thousands of different proteins that can be made by arranging these 20 different amino acids in different sequences. And again, having different sequences leads to different structure, which leads to different function. For proteins, their structure defines their function. So we often say that structure equals function. And protein structure can be very complicated. There are actually 4 levels of structure involved with the proteins. And we refer to them as primary structure, secondary structure, tertiary structure, and quaternary structure. So the first level of structure is the primary structure. And the primary structure is defined by the number and order of the amino acids. So it's how many amino acids are linked together by peptide bonds and what is their exact order. And this primary structure is going to define the overall structure, which means the overall function of the protein. So this is an example of a protein, and each individual one are the amino acids listed by their abbreviations. So glycine, proline, threonine, glycine, and so on. The secondary structure of a protein involves hydrogen bonding between molecules in the backbone of that long chain of amino acids. So hydrogen bonding, that involves those little dots, because remember, individual hydrogen bonds are very weak, but if you get a lot of them together, you get a very strong force. So hydrogen bonding in the backbone of that long chain of amino acids. And you get 2 types of secondary structure. The first is called the beta pleated sheet. And the beta pleated sheet is where you get pleats in that long chain. So pleats kind of like a fan fold if you took a piece of paper and you just folded it like a fan. The other type of secondary structure is called the alpha helix. Alpha helix, again, this is hydrogen bonding in the backbone of that amino acid chain. And this is where you get a helix, sort of like a corkscrew. So it starts twisting on itself like a spring or a corkscrew. So this is a secondary structure. And again, secondary structure is the result of the hydrogen bonds in between backbone components of the amino acid chain. Tertiary structure is the next level of structure, and tertiary structure is where this whole chain kind of folds in on itself, and it's where you get the 3-dimensional structure. So the 3-dimensional structure, and here you can see this ribbon indicates the amino acid chain, and here are some beta pleated sheets, and here is an alpha helix. And tertiary structure actually involves interaction between R groups on the amino acids. This is an example of some of the interactions between R groups you can get that influence tertiary structure. So you can also have hydrogen bonds influencing tertiary structure, but this is between R groups, not parts of the backbone. So this purple indicates the amino acid chain, and these are R groups that are coming off of the amino acid chain. You can have hydrophobic interactions, so you can have those non-polar R groups that are forced together, excluded by the water. You can have disulfide bridges that are formed between R groups that contain sulfur, and you can also have ionic bonds. So ionic bonds forming between positively charged R groups and negatively charged R groups. So these are all the different types of interactions that you can have that are going to create the tertiary structure, the three-dimensional structure of that polypeptide or that protein. The last level of structure is quaternary structure. Quaternary structure, this is where one or more polypeptides bind together to form the functional whole. So this is an example. This is one peptide structure. So that's a tertiary structure of one polypeptide. This is tertiary of another one and probably two more. They've come together to form a functional protein. And most proteins are composed of more than one polypeptide. So collagen, you might be aware that collagen is in your tissues to help hold the structure of our tissues, and it is actually composed of 3 polypeptide chains, and these are usually fibrous proteins. Kind of their ultimate structure is long and thin. This is an example of hemoglobin. So hemoglobin is formed of 4 globular proteins, polypeptides, and so altogether it is a globular protein, and its purpose is to carry oxygen to the tissues. Okay, so quaternary structure, this is where the polypeptide comes together with other polypeptides to form the actual functional protein. Many proteins in the cell and in the body are globular proteins. So they have sort of this large, rounded, globular form, and their particular shape allows them to have a particular function. So a lot of these globular proteins are enzymes, and they have a particularly shaped groove, which will allow them to catalyze reactions. One example of a globular protein is sucrase. Sucrase is an enzyme that breaks down sucrose. So it is a globular protein. It has a particular shape called the active site where the sucrose can fit perfectly in. So the sucrose will fit in there, and then the enzyme will cause a catabolic reaction, breaking that covalent bond, performing a hydrolytic reaction, and releasing the fructose and the glucose. So this is just an example where the shape of this globular protein is very important for the function. So that's again where we say structure equals function. So if anything would change the shape of this enzyme, the enzyme would no longer be able to function. Because protein structure is so important to its function, our cells have special molecules called chaperonins that ensure that the polypeptides will have the correct structure. So this is an example of a chaperonin. Polypeptide will enter the chaperonin, and inside the chaperonin it has basically a controlled environment which will allow that protein to fold correctly, and once the protein is correctly folded, then it will be released so that it can have its normal function. Denaturation is the term we use when a protein loses its three-dimensional structure. So if this is an example of a normal protein, so the ribbon represents a sequence of amino acids, and you can see that it has its secondary structure, alpha helices, that I think is a beta pleated sheet, and altogether it has its correct three-dimensional structure and maybe quaternary structure. If it encounters a condition that will cause it to lose this type of bonding, then it will denature. So it will denature into just the long sequence of amino acids. So it's lost its tertiary bonding and secondary hydrogen bonding. Now, if the conditions are not permanent, it's possible it could renature and go back to its normal shape and normal function. Some human diseases are the result of proteins having abnormal structure. And one example of this is sickle cell anemia. For sickle cell anemia, the change is in the primary structure of the protein. So remember the primary structure, this is the sequence of amino acids. So this is a normal hemoglobin gene, and in a normal hemoglobin gene, there is a glutamine at position 6. So when you have this primary structure, this leads to the secondary, tertiary structure, the quaternary structure, where you have those 4 polypeptides bound together, and that creates red blood cells of a normal shape and normal ability to carry oxygen. If an individual has sickle cell, that means they have an altered primary structure. So they actually have a mutation in their DNA. So it changes that amino acid at position 6 to a valine, which creates a completely different secondary and tertiary structure. So when this is incorporated into a hemoglobin molecule, it's an abnormal hemoglobin molecule, and now it can't function normally. And that leads to the cells having a sickle shape, or sort of a crescent moon shape, and they are not quite able to carry oxygen the way they should, and they also get stuck in capillaries and cause all sorts of problems. So this is one example of how a change in primary structure changes a protein, and that can lead to a disorder in humans. Prion diseases are another example where protein shape, or a change in protein shape, can lead to a disease. And a prion is a protein that you find in neurons, and this is an example of the normal structure of the prion disease where it has its normal function. If a person has a prion disease, that is because their prion has changed shape. So it's changed shape, and now the prion no longer has its normal function. And one example of a prion disease is mad cow. So mad cow is where a protein in the neurons folds in an unusual way, changes its function, and then that changes the function of the neurons, leading to mad cow disease. The last group of macromolecules that I want to talk about are the nucleic acids, and the nucleic acids are called nucleic acids because they were found first in the nucleus. And their main function has to do with information. So especially the DNA serves as a way to store information and transmit it from one cell to another. And also with that, most of the information that is stored in the form of DNA, we refer to it as genes, and most of the genes are storing information on the primary structure for different proteins. Now, comparing DNA and RNA in general, first thing is that DNA has a particular type of ribose, deoxyribose. RNA just has ribose. Other difference with DNA is that DNA has thymine, which I'll talk about in a little bit, and RNA has uracil. Also, DNA tends to have the structure of a double helix, and RNA tends to be single-stranded. As for their role as the molecule of information, DNA is the permanent copy of information. DNA is a very, very stable molecule. It does not break down very easily. So this holds the genes, which again holds the information for the primary sequence of proteins in general. This information can be copied into the form of RNA that forms a messenger RNA. So that messenger RNA is also holding information. The messenger RNA can leave the nucleus, interact with the ribosomes, and then that information in the mRNA is used to create the polypeptide, to arrange the amino acids in their correct sequence. RNA has a lot of different roles in gene expression, and we will talk about those different roles when we get to that chapter. But one of the things some RNA molecules can do is they can actually act as an enzyme. and those RNA molecules are referred to as ribozymes. This is an example of a ribozyme. So the structure of a ribozyme, it is a strand of RNA. And the purpose of this ribozyme is to cut or cleave RNA, so to cleave mRNA. So it's able to form a catalytic reaction and cut the RNA. So that's kind of a new area of study, relatively new, and it's one of the functions of RNA, but it's not one of the main functions of RNA. All nucleic acids are polymers that are made of similar subunits, the monomers that are linked together. And with nucleic acids, those monomers are referred to as nucleotides. And all nucleotides have the same three components. First thing they have is a 5-carbon sugar. So this is the 5-carbon sugar. Then bound to that 5-carbon sugar, they have a phosphate group. And sort of on the other side, they have a nitrogenous base. This again is the basic structure for a nucleic acid. Every nucleic acid has a sugar, a phosphate group, and a nitrogenous base. Now there are two different types of sugars that nucleotides may have. It may be a deoxyribose, which is found in DNA, or it may be a ribose, which you find in RNA. So depending on the type of sugar, that defines whether the nucleotide is found in DNA or RNA. As for the nitrogenous bases, there are 5 different types of nitrogenous bases. So there is adenine, guanine, cytosine, and then the last two, thymine and uracil. Thymine is only found in DNA, and uracil is only found in RNA. And the different nitrogenous bases are grouped into different types. The purines are the adenine and guanine. The pyrimidines are the cytosine, thymine, and uracil. When forming a polynucleotide are many nucleotides bound together. You are forming a phosphodiester bond. So linking the nucleotides together is by a anabolic reaction that forms a phosphodiester bond. And when doing so, you create a sugar phosphate backbone. So the sugar phosphate backbone, we call this because it has a sugar, phosphate, so alternating sugars and phosphates. Every backbone has 2 different ends. At one end you have a 5' end, the other end has a 3' end. And then these nitrogenous bases are sort of sticking out into the middle. A molecule of DNA is a double-stranded molecule. And with double-stranded molecule, what we mean is that one strand of nucleotides will be lined up across from another strand of nucleotides. And with DNA, the first strand of nucleotides is going to have the sugar phosphate backbone. So remember, this is sugar, phosphate alternating. And one end will be 3', the other end will be 5', and those nitrogenous bases are going to be sticking out to the center. So it could be an adenine, a thymine because it's DNA, a guanine, and a cytosine. The other strand of DNA that lines up across from the first strand is going to line up with complementary base pairing. So across from the adenine, hydrogen bonded from adenine will be a thymine. Across from the thymine, hydrogen bonded will be an adenine. Across from the guanine will be cytosine, and across from the cytosine will be guanine. So again, hydrogen bonds indicated by dots because individually they're very weak, but all together you're going to get a strong force. And the complementary base pairing is adenine and thymine line up across from each other. Guanine and cytosine line up across from each other. And again, this second strand is also going to have a sugar phosphate backbone, but it's going to be anti-parallel compared to the first one. So if this is the 5' end of that backbone, this backbone will be the 3' end, and then down here will be the 5'. So we call that anti-parallel. And that's what you see in this diagram. So the original strand of DNA, one side is the 5', the other side is the 3'. You see thymine and adenine lining across up from each other, guanine and cytosine. And then what happens is that when the DNA molecule is in water, the whole thing will twist, and that's where you get the helix, this twisting part to the double-stranded DNA molecule. Often when talking about nucleic acids, we focus mainly on DNA and RNA, but ATP is also a member of the nucleic acids, and ATP is the readily usable energy for the cell, adenosine triphosphate. Here is an example of the structure of ATP. It's very similar to a nucleotide. You have the ribose here, you have the adenine here, and then the phosphate groups. Right here, this could be an adenine. But with ATP triphosphate, you have 3 phosphates bound. And the 3 phosphates, they all have a negative charge. So this last phosphate has a lot of potential energy. Now, when using ATP for the readily usable energy for the cell, what can happen is that the ATP, the triphosphate, this last phosphate has a lot of potential energy. If it is removed, if that last phosphate is removed, then you form an ADP, which is adenosine diphosphate, and the release of energy. So that's how cells use ATP for energy. They release that last phosphate, which creates a release of energy.